In March 2020, the World Health Organization declared a global pandemic. Medical professionals and specialized scientists worked towards combating the effects of the virus. Finding the genetic identity of the virus became a top priority for the experts. That is when the use of reverse transcriptase polymerase chain reaction, or RT-PCR testing, became fundamental in decoding the virus's strain. RT-PCR testing is a laboratory technique that requires either a nasal swab sample, a throat swab sample, or even a saliva sample that is collected from the individual. In the lab, the sample gets tested for coronavirus RNA, and if present, is extracted and converted into DNA. But what exactly is the process of RT-PCR? Well, when the sample is collected, it is processed so that RNA is isolated, and every other material such as DNA polymerase enzymes, reverse transcriptase enzymes, DNA building blocks, cofactors, probes, and primers that can recognize and bind to SARS-CoV-2 nucleic acids are removed. Then, the RNA is collected and the reverse transcriptase converts it into a single DNA copy. As a result, an RNA-DNA hybrid is made. A viral enzyme is then introduced to break up the RNA and remove it from the complementary DNA. Finally, the complementary DNA is duplicated and thus creates a double strand of DNA that is now ready for amplification. PCR then works its magic and amplifies the DNA copy into millions of copies that can be readily detected. Converted viral DNA strands are then forced apart using heat and viral enzymes. When the strands cool slightly, they function as artificial sites for the DNA with primers and probes to attach, and therefore for DNA synthesis to occur. The DNA polymerase then assembles new DNA strands from the building blocks. The 5' nucleus activity of Taq polymerase degrades the bound probes, causing the reporter dye to separate from the quencher dye, generating a fluorescent signal, indicating the analysis equipment that the DNA has been successfully amplified. Chemical DNA building blocks are added and joined together in order to extend the synthetic DNA primer and form a copy of the viral DNA template. A second primer in the opposite orientation of the first primer is present in the reaction, allowing for the makeup of the complementary copy to the first synthesized strand. After the first round of DNA synthesis, the reaction mixture is heated to melt the strands apart, generating two templates ready for amplification once again. The process repeats itself and the doubling copies of DNA exponentially accumulate, eventually producing millions of copies that are used for analysis. Now, if SARS-CoV-2 complementary DNA is present in the duplicated sample, the primers smartly enough copy the targeted regions. As these regions are amplified, the probes stuck to the new fragments release a visual signal indicating the presence of the virus to the instrument analyzing the sample. The presence of SARS-CoV-2 in the genetic material then indicates a confirmed positive result. The RT-PCR testing for COVID-19 can be done in a singleplex or multiplex format. The singleplex format, or the 3 attempts format, uses 3 primer and probe sets to detect 3 regions in the SARS-CoV-2 nucleocapsid gene and 1 primer and probe set to detect human RNase P that serves to cleave off a sequence of RNA. A singleplex test can also be multiplexed into a single reaction, an amplification setup that uses 2 primer and probe sets to detect 2 regions in the SARS-CoV-2 gene and 1 primer and probe set to detect human RNase P. You may be wondering if there is a statistical difference between the result of a singleplex or multiplex testing format. A 2020 study ran testing on 93 clinical nasopharyngeal samples that resulted in both singleplex and multiplex COVID-19 RT-PCR testing methods having 100% concordance. Of the 93 clinical samples, 16 generated positive results and 77 generated negative results, using both singleplex and multiplex formats. This indicates that both singleplex and multiplex testing methods have equivalent reliable results. Over time, a virus is capable of mutating and creating different variants of its original genetic makeup. This is the case for SARS-CoV-2. Three new strains emerged in 2020, the B117 lineage, the B1135 lineage, and the P1 lineage. Although it seems intimidating, emerging variants are analyzed and immediately controlled by scientists with the aid of molecular testing such as RT-PCR. The testing is capable of picking up on different variants of the virus. For SARS-CoV-2, PCR has no issue in testing the virus's emerging variants. The PCR molecular test is designed to detect multiple genetic targets of the coronavirus and is thus less susceptible to the effects of genetic variation in comparison to a test designed to detect a single genetic target. The Center for Disease Control and Prevention confirmed that the RT-PCR test has multiple targets to detect the virus, so even if a mutation were to impact one of the targets, other targets will still manage to function. When compared to other SARS-CoV-2 tests, such as rapid antigen tests, RT-PCR remains the reference for identifying active infections. Their high sensitivity and specificity make the testing very reliable and significantly advantageous as a frontline assay. Unlike the rapid antigen tests that are less reliable as they achieve good specificity but not moderate sensitivity. Although RT-PCR molecular tests are most reliable, their capacity constraints and their relatively high cost limit their use on a massive scale. It also takes testing a longer time, from days to over a week, for results to be confirmed, in comparison to the rapid antigen testing. RT-PCR testing produces reliable results, giving medical professionals a better prediction of how many coronavirus cases they are up against.